Hey class, in this recording we're going to focus on the classification of tissues, exercise number 6 from your lab manual. So during this exercise we're going to spend a lot of time really strongly emphasizing the digital histology links. This lab has more histology, it has more astrices, right there's an asterisk, more astrices than any other lab this semester. So as we are looking at our epithelial tissues, I'm going to go ahead and pull up digital histology and just talk you through a lot of the things that we need to know for histology. First, when we look at epithelial tissues, it's important to know simple versus stratified. So all these guys up here on top, those are all simple because there's all one layer. One layer is going to be simple epithelia. And then if there's more than one layer, like all these guys down here at the bottom, if we have more than one layer, we call that stratified epithelia. And as we're looking at this epithelia, it's cool here with digital histology. I can click on the label and the label pops up for us. So I'm not going to take the time to go through and reinvent the wheel because they're all right here. So if we take squamous cells, our squamous cells right here, they have a flattened appearance. Our cuboidal cells have kind of, well, in my opinion, they kind of are spherical, but they're officially defined as having more of a, being shaped like a six-sided cube or six-sided dice. And then columnar are elongated. And something to emphasize with the columnar epithelium is that we have our apical surface and our basal or deep surface. And all the cells touch the apical surface and the deeper basement membrane. Basement membrane being this little pink line that connects the epithelial cells to the underlying connective tissues. And when we look at pseudostratified epithelium, this is kind of an in-betweeny one right here. So in this cell type, or tissue type, it looks like there's multiple layers of cells, but there's not multiple layers of cells. So every single cell still is going to touch the basement membrane and the surface layer, even though it appears that there's multiple layers. Each individual cell goes all the way to the bottom and all the way to the top, or all the way deep, all the way superficial. So from a student perspective, what's a big visual clue you can look at? I want to include, or just emphasize, the nuclei here. In a simple columnar epithelium, the nuclei tend to form a straight line from cell to cell to cell. They tend to be all oriented at approximately the same depth or elevation within the cell. And within a stratified squamous epithelium, the nuclei tend to be at multiple depths within the cells of the tissues. The nuclei are not going to form this nice, beautiful straight line. They tend to be all over the place. That's a big visual clue for differentiating the different kinds of nuclei from each other. So as we're looking at these, we talked about simple, we talked about stratified. If we were to look at this terminology we're covering, we can mix and match this terminology. So for instance, for example, let's go back to the main slide here. I could say simple squamous. That's one layer of squamous cells. This right here is simple squamous. I could say stratified cuboidal. And if it was a stratified cuboidal, it would be multiple layers of cuboidal cells. So that we actually don't even see a stratified cuboidal here. I don't like that. But right here we do have a stratified squamous. I do appreciate that about this figure. So we can at least see the stratified squamous. We also are going to have one that's kind of difficult to categorize. That's the transitional epithelium. And as we look at our transitional epitheliums, those are going to have the scalloped edges or the bumpy edges to the apical surface or surface layer of cells. And it's also going to be multiple layers of cells thick. I'll spend some time pointing that out to you. As we look at the components of our tissues, we have goblet cells, cilia, and lumen. A lumen is a hollow space within a tubular organ. A cilia is a small hair-like extension that comes off of a cell. We can see cilia being represented right here as these little hairy extensions within this figure, but that's not very helpful. Let's pull up some actual histology to show you that stuff. Let's go with Histology Guide. And I want to show you some ciliated stuff right here. The larynx has a bunch of ciliated tissues. So here within our larynx, we have a pseudostratified columnar epithelium. So this layer, this band of cells right here that I'm drawing a big green box around, this is a pseudostratified columnar epithelium. It looks like there's multiple layers of cells, but there's really only one. Let's go in red here, and with that red arrow, I'm pointing towards those cilia that are sticking out from the cells, those small hairy extensions. And then let's go with yellow here. Oh, I did not want that. Let's go with yellow here. And in yellow, I'm circling. Let me get my pen back up. Here we go. I'm going to go ahead and circle these clear patches. These clear patches represent goblet cells, or the mucus makers. A goblet cell is a mucus maker. It's going to produce the mucus that makes up a lot, that lines our airways, our respiratory tracts, and our digestive tracts. And if we look at our, let's go ahead and exit out of full screen and go back to our lab objective sheet. That was the goblet cell, cilia, and the lumen I didn't cover. Let's pull this back up. The lumen is the hollow space. So this is from our trachea. So this, actually, let's back up. We can go with a better image here. This is a fantastic image. So here's the esophagus, a transverse slice of the human esophagus. And right here, in the middle of our esophagus, we have a bunch of empty space that I'm drawing this red line in. This empty space in the middle of this tubular organ is called a lumen. It's a generic term that'll appear over and over again. We have a lumen in the small intestine, a lumen in every blood vessel, a lumen in most of our ducts and our glands, or our duct-based glands. So we have lots of lumens. As we're looking at our epitheliums, we have simple and stratified epitheliums. I'm going to go ahead and show us some epitheliums right now. So let's start with our simple epithelium. We have a simple squamous epithelium. And as we're looking at the simple squamous epithelium, I want to emphasize to you that we need to focus on just the surface layer of cells. So right here that I'm outlining in green, we have a simple squamous epithelium, one layer of flattened cells. And we can see individual nuclei of those simple squamous epithelial cells right here that I'm drawing those little purple or the green arrows towards. And the simple squamous epithelium goes all the way around this cavity or this space. As we look at the simple squamous epithelium, it's one layer of flattened cells, and it does a really good job of forming a barrier or a membrane in our bodies. Oh, wrong button again. Let's pull up a simple cuboidal epithelium. And within our simple cuboidal epithelium, let's go for just this slide right here, simple cuboidal. There's a lot of simple cuboidal epithelium in different organs of our body, including the kidneys, around the renal tubules. And as we look at these cells that form the simple cuboidal epithelium, I want to emphasize that we're focusing on just this surface layer of cells. And note how there's many, many dark purple nuclei closely packed together, and each individual cell is roughly cuboidal in shape, hence the name cuboidal epithelium. And since there's only one layer of them thick, it's a simple cuboidal epithelium. And as we're looking at simple columnar epithelium, I haven't shown you that one yet, so let's go ahead and pull that up. We have simple columnar epithelium. Let's go with the, I'm used to using the stomach, but we can use the small intestine too. The cells that line the small intestine are simple columnar epithelial cells. So when we're looking at this, we can see how the cells are elongated and how the cells have all of their nuclei at approximately the same height, and how, as we look at these cells, the cells themselves are only one layer of cells thick. So because it's one layer of cells thick, we call it simple. Because all the nuclei are approximately the same depth and the cells are elongated in shape, we call it columnar. So this is a simple columnar epithelium. And I would be remiss if I didn't highlight these goblet cells. So there's these big clear patches right here in the tissues. These are all individual goblet cells. And this individual right here, where I'm drawing that arrow, you can see the strands of mucus being vomited out into the lumen of the small intestine where this slide was made. That's pretty cool. We also have stratified epithelium. And as we're looking at stratified epithelium, we're going to go look at digital histology. I want you to focus on stratified squamous epithelium and transitional epithelium. Yes, there are stratified cuboidal epitheliums and stratified columnar epitheliums in the body, but they're really hard to find. There's not many of them. So we're going to focus on our stratified epithelium. We'll first focus on stratified squamous. So in the esophagus, we have non-keratinized stratified squamous epithelium. Here in this lavender purple color, you see how there's many, many layers of cells thick. And as we're looking at these cells, you can see how they are elongated and flattened out. And because there's multiple layers of these cells, that makes it stratified. The flatness or elongation makes it squamous. And when we're looking at a stratified epithelium, we always are going to name it based off the apical layer or surface layer of cells. So right here, facing the lumen, this is the apical layer of cells. And these cells in the apical layer are going to be different looking from the deepest layer of cells down here by the basement membrane. The apical layer of cells are all flattened out. The basement membrane cells have not yet been flattened out. But since we only look at the apical layer of cells for naming the morphology or shape, we say this is a squamous epithelium, and because there's multiple layers of cells, it is a stratified squamous epithelium. That was non-keratinized, though. Let's go ahead and show you some keratinized stratified squamous epithelium. Come on. Oh, I zoomed in on the wrong spot. Double click. I'm used to the double click centering my field of view. So here's a keratinized stratified squamous epithelium of thin skin. And here, this apical layer of cells are totally flattened out. They look like little flakes, or they're sloughing off. So here at this apical layer, they are completely keratinized. They no longer have a nucleus. This is fully keratinized stratified squamous epithelium. So depending on whether the cells have been filled with that tough waterproof protein keratin or not, we have a difference here. Keratinized versus non-keratinized is going to change the appearance, but they're still all stratified squamous. And then finally, we have our transitional epithelium, also known as urothelium epithelium. That's just awkward sounding to me. I'm a little crustier and older. I'm more of a transitional epithelium kind of guy. The reason urothelium is becoming popular, and I shouldn't say becoming popular, the reason urothelium has been introduced is because transitional epithelium is only in the urinary tract. So because it's only in the urinary tract, it might, kind of make sense to refer to it as a urothelium. So right here, this is a non-distended transitional epithelium, and this spot right here really emphasizes that scalloped edge that I had mentioned earlier of the transitional epithelium. So here is the transitional epithelium, non-distended. And a unique feature about the transitional epithelium is that it allows for the tissues to stretch out. So over here on the left, we have non-distended transitional epithelium. Notice the scalloped edges. And then on the right, over here, it's been stretched out. We no longer have the nice scalloped edges. This is distended transitional epithelium. So that was it for epithelial tissues. I'm going to call this video quits because it was a bit long already. We'll pick up with connective tissues with our next recording. If you have any questions or comments about epithelial histology, please feel free to post them in the class discussion boards or to shoot me an email. And as always, happy study.